In continuation to Oomycota, the last one is comparative differences. Now we will see what are the different comparative differences that we can expect from Oomycota and Plasmodiophoromycota. Since we have already finished Plasmodiophoromycota and Oomycota, now we will see the comparative differences in these two phyla. I'm N.H. Shankar Reddy, PhD Plant Pathology. So let's start with the difference between Oomycota and true fungi. So here actually Oomycota is a fungi-like organism. It's not a true fungi. That's why this Oomycota actually placed under a lower fungi. So when we see higher fungi like Ascomycota and Basidiomycota, comparatively to higher fungi, this lower fungi have they exactly look like fungi only, but they are not true fungi. So now we will see the what are the general differences that we might observe, in Oomycota and true fungi. So here I divide into two, that is a Oomycota and true fungi. So like Ascomycota or whatever the Basidiomycota. So now see the general comparative differences. So other names generally, this Oomycota is also known as the egg fungi, are also known as the pseudo fungi. The reason is because in Oomycota they produce a sexual spore as a oospore, right? So the oospore is look like an egg. That's why it's called as egg fungi. Why it is called a pseudo fungi? Because it's not a true fungi, it's a fungi-like organism. That's why it's called a pseudo fungi. Pseudo means not true, like a false, we can also say. That is true, pseudo fungi. So every name behind there might be a reason also that we, if we study in that dimension, it will be very easy for us to remember, I think. So anyway, true fungi like true fungi, generally true fungi only, like Ascomycota and Basidiomycota. So I have mentioned sac fungi. Why it is called Ascomycota is sac fungi? Because the ascus actually produced in a sac-like structure is called. They produce ascoma like they produce a sac-like structure. All the Ascomycota group, especially mushrooms, they produce a sac-like structure, and almost all ascospores are produced inside a sac-like structure called ascus. So inside the ascus, ascospores are there. That's why it's called as a sac fungi. And we can also call Ascomycota as a crown fungi because it is one of the largest phyla where majority of the fungi was grouped under, right? And Basidiomycota generally called as a club fungi, where basidiospores are produced above a club-like structure. That's why it's called as club fungi. So this is other names. Cell wall, generally if you see Oomycota group of cell wall is made up of cellulose. We know very well that fungal cell wall is made up of chitin, a little amount of glucan as well. But when it comes to Oomycota cell wall is made up of cellulose and a little amount of hydroxyproline. It's a very, very important. In exams we expect like generally Oomycota cell wall is made up of cellulose, or sometimes they may give other options like hydroxyproline. In 2023, I think this 2021 probably this question was asked, I think 2021 or maybe 2023, I'm not sure, but this question was asked. They gave like Oomycota fungal cell wall is made up of, they never have mentioned an option cellulose, but they mentioned hydroxyproline. So in such a way also we may expect questions as well. But when it comes to here, generally Oomycota cell wall is made up of cellulose and hydroxyproline, well as true fungi like Ascomycota, Basidiomycota, majority of the fungal cell wall is made up of chitin only. When it comes to Zygomycota, chitos and chitin, but when it comes to true fungi, majority of the true fungi cell wall is made up of chitin. So cellulose and hydroxyproline is the chief ingredient when it comes to Oomycota. But there are couple of fungi, even though they comes under Oomycota, their cell wall is made up of chitin. For example, fungi like Acaila, Aphadogyla, Saprolignia, and Leptometus. So such there are exceptional fungi, even though they comes under Oomycota, their cell wall is made up of chitin. But generally Oomycota group of, for example, Pythium, Phytophthora, all those things generally comes under Oomycota, right? Their cell wall is made up of cellulose only. Okay. And storage organ, food storage organ is in the form of mycolaminarin, whereas in true fungi, the food storage or reserve food material in the form of glycogens or oils or fats, like a reserve food material. However, we have adipose tissue for humans. Like that fungi also they have storage organs or like how can I say, like food storage products in the form of mycolaminarin and Oomycota, whereas in true fungi glycogen and oil are fats. Flagella, generally Oomycota fungi produces flagella, which are motile. That's why it's called the juice pores and all those things we used to discuss in Oomycota group of fungi in the earlier, right? So whereas in true fungi, they produce conidia. For example, Ascomycota and Basidiomycota produce some kind of conidia, which are non-motile. Why it is non-motile? Because it contains no flagella. But when it comes to motile, they contains flagella. For example, you can see Oomycota group, the flagellation is anterior tinsel and posterior whiplash type of flagella we used to discuss there, right? In flagella also we have a very great explanation about this kind of flagella. Especially Oomycota group of fungi are highly famous for the production of flagella. Juice spores are generally flagella. So if it is the spores are motile, it is called as juice spores or planospores. It's a non-motile, you can call it as aplanospores, right? like that. Hyphae generally cynocytic. Cynocytic hyphae is also known as non-septate or aseptate hyphae. So there is no cross walls or there is no septa between the hypha. When it comes to true fungi hyphae is generally septate. Is actually a well adapted, very good adaptation. Having septa is a well good adaptation. For example, sometimes if the protoplasm was moving or protoplasm was completely shifted to one direction, the other side of the hyphae might be in the risk or the chances of death, right? So to prevent that, higher fungi like Oomycota and Basidiomycota, they have septations. But anyway, this lower fungi also having well adapted things, even though they don't have septa. But generally cynocytic means no septa in the hyphae, septate means septa in the hyphae. Sterol product fucosterol, it's also very important. The type of sterol compound present in the Oomycota, fucosterol. When it comes to true fungi, ergosterol. So sugar and alcohol generally absent when it comes to Oomycota. That kind of stuff are present in true fungi. Asexual spores are juice spores. Just now I told you motile spores are juice spores, right? And mostly conidia, which are non-motile. And sexual spore is oospore. Oospore is a very thick-walled resting spore that is produced during unfavorable time. And in fact, to survive, the Oomycota generally produces sexual spore is known as oospore. They are very thick-walled and very thick walled and like diploid in condition. They survive in soil during unfavorable time, so they look like egg. That's why it's called as egg fungi. Okay, and generally this true fungi, Ascomycota group of fungi produces ascospore, whereas Basidiomycota group of fungi produces basidospores. Both spores are haploid. When it comes to zoospore, oospore is a diploid. Okay, so oospore is a diploid resting spore, whereas ascospores and basidospores are haploid and non-resting spores. When it comes to flagellation, generally flagellation is present. That's why we discussed, right? Juice spores are generally biflagellated with anterior tinsel and posterior whiplash. I will tell you what is anterior tinsel. This is flagella. Tinsel means flagella having hair. So this small hair will be there. That is called fibrils, or we can also name as masticoniums, right? And there, if the flagella having no flagella, sorry, the flagella having no hairs, it is called whiplash. That's what it's called anterior, because the flagella is anterior direction. Anterior tinsel, tinsel means having flagella. Posterior means downward direction. Whiplash means flagella having no hair. So anterior tinsel and posterior whiplash, that is a very important character of Oomycota fungi. And here this higher fungi very rarely produced. If produced, maybe one or sometimes there is no flagella will be then. If it is present, maybe posterior whiplash type of flagella, and mostly rare. It is very rare when it comes to true fungi, but when it comes to Oomycota or lower fungi, it's very often. And sexual reproduction is oogamous by means of gametangel contact. We know very well this is oogonium, this is antheridium, where they come in contact with each other. So the type of reproduction in Oomycota is pure and pure of gametangel contact, where male gametes like antheridium and female gametes like oogonium come in contact with each other and exchange a nuclei, right? And all those things we have discussed in the previous lectures as. So anyway, just like recalling all those things. Anyway, the gametangel contact type of sexual reproduction can be observed in Oomycota. When it comes to true fungi, again gametangel contact, gametangel copulation, rarely spermatization, and somatom, all those things that we can observe in true fungi. That is Ascomycota and Basidiomycota. And anyway, lysine pathway, it is also very important. I think this question was asked in general kind of exams, like, so the kind of pathway that we can observe in lower fungi like Oomycota or Pythium, Phytophthora. Sometimes they also mentioned this fungi for confusing us. Okay, generally for lower fungi or Oomycota, they're having a diaminopimlic acid pathway. When it comes to true fungi, they're having alpha amino adenic acid pathway, adipic acid pathway anyway. Okay, so the lower fungi having a diaminopimlic acid pathway, whereas higher fungi like Ascomycota basidium having alpha amino adipic acid pathway. Best examples that comes under Oomycota is, a couple of examples are there, like Pythium, Phytophthora, downy mildews, all the downy mildews that comes under Oomycota group of fungi, and true downy mildews. Okay, and when it comes to true fungi, most important thing is the rust, smut, mushrooms, I would say powdery mildews, other things are there, and all those things are comes under this true fungi. And the next important thing is the next difference between Pythium and Phytophthora. This is also very important. We mostly expect this question in not only in semester exams but also in various other kinds of exams like ARS and all those things. Anyway, let's see the difference between Pythium and Phytophthora. When it comes to Oomycota, other fungi is also there, but the most important concentration will goes to two fungi, that is the Pythium and Phytophthora. Especially these two fungi are very important when it comes to Oomycota, and downy mildew is also there, that is secondary, but let's concentrate on this Pythium and Phytophthora. So this Pythium generally causes various kinds of disease, damping of very well. That Phytophthora we know very well, that lead blight of potato, one of the remarkable disease. Without lead blight of potato there is no pathology, without pathology there is no me, right? Just jokes apart. Anyway, so let's see Pythium. Meaning of Pythium is generally Pythium means, the general meaning is decay. Phytophthora means plant destroyer. Phyto means plant. Thora means destroyer. Generally, Phytophthora means plant destroyer. It was named by Anton de Berry because it's destroying the entire plants. So that he named as Phytophthora. Phyto means plant. Thora means destroyer. Phytophthora means plant destroyer. So generally, this Pythium comes under Pythiaceae before. Now it is changed into Peronosporaceae now it comes under. So this Phytophthora comes under Peronosporaceae. Order of Pythium is Pythiales and order of Phytophthora is Peronosporaceae. Now this Pythium is also placed under Peronosporaceae and Peronosporaceae. So the previous classification is it is under Pythiaceae and Pythiales, and now it's the present classification is under Peronosporaceae. If you want, you can also just go and check the present classification. Now it is classified under Peronosporaceae. Generally symptoms, Pythium, very common symptom is just decaying of roots, or like rotting of roots, or stunted growth, or some kind of death before or after germination of the seedlings from the soil region. So that is a very important symptom of Pythium. When it comes to Phytophthora, it will produce some almost more or less similar kind of symptoms, but with light variations. Generally rotting of stems or discolorations or wilting of plants, or some kind of water soaked lesions or discolorations on the leaf. If you see the potato, like decaying of potato, we can observe such kind of symptoms. So generally parasitism is this Pythium is a best example of facultative parasite, and whereas Phytophthora is a facultative saprophyte. Haustoria and Aprosoria absent. It is also very, very important, right? Pythium and Pythium never produces Haustoria and Aprosoria. In very rare cases, some people reported it may be produced. As far as my an observation on the lots of literature I have gone through, many people mentioned that. I have no direct experience, but I'm generally telling. So by having lots of review from the various literature, what I found is majority of the literature mentioned that the Pythium produces no Haustoria and Aprosoria. Very few literature found, maybe a couple of species may produces. If you guys know, please let me know also. I will be very happy for that. And anyway, Haustoria and Aprosoria, generally this Pythium produces no Haustoria and Aprosoria. That is also one of the specialty for this Pythium. And the Phytophthora generally produces Haustoria and Aprosoria. We know very well this Phytophthora known to produce a finger-shaped Haustoria. That is also one of the important feature of this Pythium. And sporangiospores generally not well distinguished from hyla. I repeat this point. These sporangiospores are not actually well distinguished. There is no well distinguished from hyphae. They look like the same, but there is no well distinguished. But when it comes to the sporangiospores of Phytophthora, it is a very well distinguished, and the branches also like zigzag branches are sympodial branches like a tree. So very well distinguished when it comes to the sporangiospore of Phytophthora, but it is a little bit difficult to differentiate this Pythium. Sporangia generally look like a spherical or ovoid shape, whereas the sporangia look like lemon shaped. This is also very, very important. And the sporangial position is always the Pythium was actually born on either terminal or intercalary, but when it comes to Phytophthora, they born on terminal position only. Hyphae generally narrow hyphae. When it comes to Phytophthora, it is a little bit wider hyphae compared to the Pythium. And generally, the nature of growing is fast growing. I know, I have direct experience because a couple of friends who are working in my MS and PhD, they were working on Pythium. Generally, Pythium grows very fast. Like if you start to culture on the petri plate, maybe it will take just 2 to 3 days, like a sclerotium. It will cover very fast, and it will look like some kind of house fungus, like some kind of fluffy growth we can observe. Anyway, this is very fast growing when compared to Phytophthora. Phytophthora is a little bit slow growing. Maybe as far as my experience, may take around 7 to 14 days to have a full covered plate, and sometimes also take a little more time based on the temperature as well. And vesicle, very important point, formation of vesicle above the sporangia is a very important character of Pythium. For example, this is a sporangia. So in the life cycle of Pythium also we have discussed about this Pythium, a special character. This generally Pythium produces not only Pythium but also Phytophthora produces sporangia. Above the sporangia, this Pythium produces another structure called vesicle. In the vesicle only juice spore formation or compartmentization will start. Actually, when it is in the sporangia, generally it is like a state like a protoplasm. When it starts to move into the vesicle, where compartmentization takes place and the juice spore production takes place in the vesicle only. So the formation of vesicle is one of the special feature of this Pythium. So when it comes to Phytophthora, there is no such kind of forming of vesicle and all those things. Okay, release of juice spores, whereas the juice spores are released in a rocking motion when it compares to Pythium. Generally the Phytophthora puffing mechanism. So differentiation of juice pores are actually, as I told you, juice pores are actually differentiated, or differentiation or formation of juice pores only takes place in the vesicle. So when it comes to the Pythophthora, there is no special formation of vesicle, so that the juice pore formation takes place in the sporangia itself and inside the sporangia, and they are released whenever they get matured. Okay, and oogonial wall generally brownish in color. They never be spiny. When it comes to Phytophthora, oogonial wall is hyaline and a little bit smooth. Antheridia, here it is also very important. The type of Antheridia in Pythium is a paragynous. So paragynous means where a male reproductive organ, Antheridium, and female reproductive organ, oogonium, they lie side by side. That is called paragynous. So if it is ampigynous means actually uh, male reproductive organ passes through oogonium, that is the female reproductive organ, and form a globo structure above the female reproductive organ passes through the antheridium and they form a globo structure above the antheridium, which means this is the antheridium, and this oogonium actually passes through the antheridium and they form a globo structure above the antheridium. So such kind of antheridium is known as ampigynous type of antheridium. That is the special feature of Phytophthora. And generally baiting technique for this is isolation. We do recommend, not me generally. So the Pythium can be isolated by using mustard seeds, and when it comes to apple, that is recommended for isolation of Phytophthora. Best examples are Pythium aphanidermatum, generally causes damping of vegetables, and Pythium debarium other species. Generally damping of disease this Pythium causes. When it comes to Phytophthora, the best example is lead blight of potato. Hundreds of species are there. I think in the previous presentations, when it comes to the Peronosporaceae, we have clearly mentioned what are the list of diseases that is caused by Pythium, what are the list of diseases that is caused by Phytophthora. This is like just differences. So there are very important points we have covered in this, like formation of vesicle is a special feature of Pythium or Phytophthora. Pythium or ampigynous type of antheridium is present in Phytophthora. Such kind of questions only we do expect in not only net exam but also in various kinds of exam. If we study in this way, it will be very easy for us to remember the stuff, and in the meantime, not only for immediate preparation but also for semester and various other kinds of exams as well. Now we'll see the difference between Pythiaceae, Peronosporaceae, and Albuginaceae. These are all actually comes under this Oomycota only, right? So Pythiaceae, Peronosporaceae, and Albuginaceae. Generally habitat Pythiaceae are saprophytes, right? And these are also saprophytes. Couple of them are Peronosporaceae are known to be obligate parasite, but Albuginaceae is obligate parasite. And Sporangiospore and Conidiophores. I think we have already discussed this. Pythiaceae generally there is no such kind of differentiation. They are undifferentiated from the mycelia, and they are branched intermediate growth, and they produce sporangium or conidium, generally sporangium only Pythiaceae, right? And when it comes to Peronosporaceae like Phytophthora, they have a well differentiated mycelium. I mean, they actually differentiated from the mycelium, and they actually emerges as a singular, sometimes maybe tufts like branches, and with a determinate growth, and there is no subsporangial growth can be observed in this Peronosporaceae like of Pythiaceae. And Albuginaceae, generally the sporangiospore is clavate, look like a clavate, unbranched. And the other feature of this Albuginaceae is chains of sporangia can be observed, like this is also I think we have discussed in Albuginaceae. The specialty of this Albuginaceae is they produce a knob-like hastoria. First thing, very important, knob-like hastoria. The second thing, they produce chains of sporangia. But when it comes to powdery mildew produce chains of conidia. But when it comes to albuginaceae produce chains of sporangia, and they are very dense and like a cluster kind of thing that we can observe in Albuginaceae. I think we have discussed in Albuginaceae in detail about this Albuginaceae and albugo disease caused by albugo along with the life cycle we have already discussed. If you guys have not gone through, we suggest to go through. And anyway, last one is a periplasm. So oogonial periplasm is either present or absent in Pythiaceae. When it comes to Peronosporaceae, it is very well developed periplasm when it comes to the group of Peronosporaceae. And Albuginaceae, that is persistent and conspicuous. And Hastoria, as I told you, Hastoria and Aprosoria is not present. Maybe some species it may be chances of present. But Peronosporaceae generally it clearly produces Hastoria, especially Phytophthora to produce finger-like Hastoria. And as I told you, Albuginaceae another special feature is a knob-like Hastoria. It is also very important. Knob-like Hastoria is produced by this Albugo candida and albugo species generally. This is caused by this albugo, that is Pythiaceae is the damping of vegetables, Pythium aphanidermatum, and Peronosporaceae we know very well lead blight of potato, right? Albuginaceae white rust of mustard, albugo candida. These are all the different kinds of diseases caused by. And the next one is the difference between Plasmodiophorales and Peronosporales. General difference only. So this Plasmodiophorale general comes under Protozoa, whereas the Peronosporales comes under chromista, right? The division kingdom. So phylum of Plasmodiophorales is Plasmodiophoromycota, whereas Peronosporales is Oomycota, right? Thallus is generally Plasmodium. The Plasmodium multinucleate mass of protoplasm is known as Plasmodium, right? So when it comes to this here, thallus is not Plasmodium. We have very well developed thallus here. There is presence of septa or asepta, that is secondary, but it has very developed. I mean, here in the Peronosporales, it is thallus is present. When it comes to Plasmodiophorales, thallus is a Plasmodium. So Plasmodium itself is a thallus. Okay, na? Sexual spores is like the resting spore. Generally, this Plasmodiophorale produces cyst, whereas in Oomycota we know very well oospore is a sexual spore. Asexual spore in Plasmodiophorales is zoospores, whereas in asexual spores in Peronosporales is zoospores and conidia. Cruciform type of divisions are present. So continuous type of divisions. In the life cycle of Plasmodiophorales, we have discussed what is cruciform or cruciferous or cruciform type of divisions, are continuous kind of replications or divisions in cruciform type of divisions that we observed in Plasmodiophoromycota. Such kind of divisions are absent in Peronosporales. Peronosporales are nothing but generally this late blade of potato and all those things only. Okay. In Plasmodiophorales, we have only one fungi, clubroot of cabbage. Nothing to worry about it. You just compare to although the two fungi like clubroot of cabbage, Plasmodiophoromycota, and this Phytophthora species. If you compare this, the difference will be very easy. Type of zoospores is anteriorly attached in types of flagella. So in types of flagella, Plasmodiophoromycota produces anterior whiplash type. One is short, one is long, that we have discussed in the types of flagella. So when it comes to Peronosporales, anterior tinsel and posterior whiplash. Biflagellate with anterior tinsel and posterior whiplash like this. Anterior tinsel contain hair, posterior whiplash no hair. That is a special feature of all Oomycota. In fact, Peronosporales comes under Oomycota, right? So like that. Sexual reproduction is planogametic copulation, whereas here gametangial contact when it comes to Peronosporales. So vector for viral disease. Here it is a little bit important. Some of the species that comes under this Plasmodiophorales like Polymyxa, Spongospora act as a vectors in transmitting of plant viral diseases. And here when it comes to Oomycota or true fungi, there is no species have been involved in transmitting of any plant viral diseases. But this Polymyxa and Spongospora are very very important. For example, Spongospora subterranea, PVX, Protovirus X, Polymyxa graminis tuberculosis virus. So such kind of diseases it is used to involved in transmission. Parasitism when it comes to Plasmodiophorales, it is an endoparasite. When it comes to Peronosporales, some species are obligate parasite and also saprophytes also there. And other names parasitic slimy molds. And when it comes to other names of Peronosporal, zoosporic fungi because they produce zoospore, right? Pseudofungi because it's not a true fungi or fungi-like organisms. Because it is a slimy mold, that's why it is a Plasmodium. So Plasmodium like a slime, right? That's what comes to not that is the exact reason, but generally telling for our understanding. So example for Plasmodiophorales, I told you clubroot of cabbage, very very important. Spongospora subterranea, potato scab. Actually this Spongospora comes under other group, but anyway more or less similar character, that's why I placed it here. And anyway Peronosporales, very important one is late beta potato, white rust of crucifers, and all the downy mildews comes under true downy mildews comes under this Peronosporales. These are all the comparative differences. For more information, we suggest to take a look into this book, A Vision into Plant Pathology, complete student version. For further doubts, clarification and guidance, students can reach us at www.geekyresearcher.com. Stay geeky and stay tuned. We are Team Geeky Researchers.